Hello, my name is Martin Blaser. I'm the director of the Center for Advanced Biotechnology and Medicine at Rutgers University. Today, I have the pleasure of interviewing Dr. Conor McClenaghan, who is a new faculty member at CABM, as the center is called. Conor is an assistant professor in the Department of Pharmacology, and he is also a resident member of CABM. So welcome, Conor. It's great that you're here. And so I just want to ask you a couple questions for our uh, large worldview audience. So I guess the first thing is you're a scientist. Tell me about your initial interest. How did you get interested in science? And tell us a little about your early education.
Yeah, sure, Marty. Thanks for speaking today. Thanks for the invitation to chat. As you might tell already, I'm from England. So my early education, of course, was in England. I grew up in a part of the country called Cheshire, and I went to do my undergraduate degree in pharmacology at the University of Leeds, which is in Northern England. And actually, it was there that I first turned my hand to research in a final year thesis project, which I think was 6 or 8 weeks of work. And actually, during that project was when I first learned the experiment, which I'm still doing all these years later. So in that project, I was introduced to electrophysiology. So this is a series of techniques to explore electrical behavior in the body. And so during that research project, I learned a process called patch clamp electrophysiology, which I remember vividly hating at the time. It was such hard work, and I kept failing and kept failing. I was being trained by a great teacher called John Lippiat, who's an expert in potassium channels. But despite his attention, I was really struggling. So I remember at the end of that project thinking, never again. I'm never doing electrophysiology ever again. And that was about 15 years ago. So I'm still at it now. So I got some things wrong, but it worked out quite nicely. So after Leeds, I worked in industry for 3 years for Novartis on some drug discovery projects. And then I did my PhD in England at Oxford University, again focusing on potassium channels in under the supervision of Stephen Tucker in the Department of Physics. So there we were trying to understand how these channels look and how these channels move when they are activated and when they close.
And then you did a postdoc as well.
Right, correct. So that's what brought me to the States. After my PhD, I moved to Washington University in St. Louis to undertake a postdoc under the mentorship of Colin Nichols, who is a world-leading expert in ATP-sensitive potassium channels. So this is a different family of potassium channels to those that I worked on previously. So I've not ventured incredibly far scientifically, but now looking at a different element of these channels. So we're trying to understand in the postdoc, we were trying to understand the consequences of the dysfunction of these ion channels for human disease. And that's really something which I'm continuing in my own lab now.
Well, and so how long have you been at CABM now, and what brought you here?
Yeah, great. So I started at CABM on the 1st of June 2022. So it's just
Ah,
past the 1 year birthday for the lab. So it's
congratulations.
a timely conversation, yeah. So I've been here, yeah, just over a year now, and really I was drawn to Rutgers by obviously it's it's a renowned reputation for research, and particularly at CABM by the breadth of the research that's going on here. So it's a really fascinating institution with a wide range of different research directions all encompassed under this one roof. So that was very exciting, and I was sure, and I have been vindicated in expecting that I would be exposed to different kinds of science here. And so I really think in science in general, we can make our, surprising advances when different disciplines intersect. So there's a real potential for that in this building. And then there's many faculty members with very successful careers, which young faculty like I can hope to emulate and try and learn some things from. And then on top of that, it's a really welcome and friendly environment as well. So I've been really excited since I've been here.
And we can see that they also give you a lot of shelf space.
Right. Yeah, I don't know
Great.
whether in the modern world when we don't have many books anymore and
You
everything is
know, they don't
digital.
have to be books, you know, they
Right.
can be your artwork. Yeah.
Exactly. I need some keepsakes now. Yeah.
So then what are you working on during this 1 year and a couple of days?
Right. So yeah. So the main focus in the lab is continue to work on a family of potassium ion channels called ATP sensitive potassium channels. So ion channels are proteins in the body, and they reside in the membrane of cells. So the membrane obviously is this lipid layer that surrounds all cells in the body. It keeps the intracellular components within the cell and protects the cell from the extracellular components. And many people have the understandable notion that there is electrical activity in the body, but maybe you think it's confined to the brain or the heart, which are known to be electrical organs. But actually, all cells in the body can include electrical signals. So these are very small currents which pass across the membrane, and these currents pass across the membrane through these ion channel proteins. So these are tunnels which open and close to allow the movement of ions in and out of cells. And ions are charged atoms, and therefore they carry a current in and out of the cell. And so it's the movement of these ions which underlies the electrical behavior in different cell types. And this electrical behavior is really coupled and controls many other cellular processes. So we're at a fundamental level, we're trying to understand how electrical behavior regulates different processes in the body. So our particular focus is on these ATP sensitive potassium channels. So these are potassium channels. They are responsible for allowing the movement of potassium across the membrane, and they are regulated by intracellular ATP. So ATP, as you know, is an energy reserve within cells, and I suppose we can think of it as cash in the bank. So when a cell is doing well, it's well fed, it's well oxygenated, it's capable of producing reserves of ATP, and this ATP can be used for cellular transactions and for fueling reactions within the cell. And so this is a vital reserve within the cells. But if a cell undergoes metabolic stress or hypoxia or reduced perfusion, let's say, ATP levels can be depleted. And this is particularly true in certain metabolically active cells like neurons or muscle cells. So when a muscle cell is working, it uses up its ATP stores relatively rapidly. The KATP channels sense this decrease in ATP and open, and this allows for the potassium within the cell to leave the cell down its electrochemical gradient, and this controls the electrical behavior of the cell. The activation of potassium channels in muscle in general leads to relaxation of muscle cells, and this can preserve energy levels within the cell.
So let me just stop you there. So what's what's a key question that you're working on?
Right. So with all of this knowledge about what these channels do in general physiology, what we're really interested in is how these channels, what are the consequences of these channels dysfunctioning in disease? And so one project that we're actively working on is what are the consequences of genetic variations found in the genes which encode for these ATP sensitive potassium channels in human diseases. And so we're currently working on 2 different diseases, and one is caused by mutations which cause overactivity of these channels, and another disease is caused by mutations which cause loss of function or underactivity of these channels. And so we're currently trying to work out how do these mutations lead to quite diverse clinical features in people who carry these mutations, and of course, ultimately, whether we can correct the pathology that's observed in these people in the future by targeting these channels with therapeutics.
Well, so what are the diseases where you have over and under activity?
Right. So the overactivity syndrome is called Cantu syndrome. So this is a rare disorder. There are around 150 documented cases so far, but that's likely a severe underestimate. There's probably tens of thousands of people worldwide. And in that disorder, the disorder is characterized by distinctive facial appearances and excessive hair growth in people. But alongside those dysmorphic features, the people also present with a wide range of cardiovascular abnormalities. And so some of the work we've done so far is trying to understand how those cardiovascular abnormalities arise and how they can be reversed once they're present. The loss of function disorder is a bit of a mouthful. This is called ABCC9 related intellectual disability and myopathy syndrome, or AIMS, AIMS for short. And so, as the name suggests, this is a neurodevelopmental disorder with intellectual disability quite commonly observed. And patients also exhibit cardiovascular abnormalities as well as skeletal muscle dysfunction. So they fatigue quickly and they describe episodes of painful muscle cramping during exercise.
Yeah. So you're going from genetics all the way to clinical medicine. So at what level are you approaching this problem?
Right. So as you say, yes, we're trying to understand how do these mutations in the genetic code change the function of the protein at a molecular level. And so we express proteins in cell lines and we measure the behavior of the ion channels using electrophysiological techniques in recombinant expression systems. So in cell lines, in vitro, in the lab. And then we also have genetically modified mouse models where we've introduced the equivalent mutation, which we find in humans, into the KATP channel genes in the mice. And what we found is that these same mutations cause all the same cardiovascular abnormalities in mice that we see in humans. So now we can use our in vivo models, our mouse models, to try and determine how this pathophysiology arises. What are the systemic mechanisms by which the pathology arises? And then ultimately whether we can reverse them.
So, of course, it all sounds pretty good to me, but to you, what's a particularly exciting project you're working on?
Right. So the loss of function disorder was only relatively recently identified. So we were part of an international collaboration with many clinical colleagues who initially found two families who both had the same mutation. And these families were found in northern Norway. And so that was the first identification, clear identification to my mind, of a loss of function mutation in this gene, leading to a clear human pathology. And so with that, since that first report 3 or 4 years ago now, we've identified many more patients with different mutations. They all lead to the same loss of function of the KATP channel activity, and they all lead to the same clinical constellation of pathology. So we're now pretty confident that this genetic syndrome is clearly caused by loss of function of KATP channels. And this is interesting and exciting because if we know the gene that's responsible, we know the protein that's responsible, and therefore we have a target to try and correct it. So what we're currently trying to do is to find ways in which we can replace the faulty gene with a functional gene and maybe rescue channel function in our animal models, and that would be the first step towards potentially identifying a clinically viable therapy for these patients.
Yeah, sounds exciting. I guess we'll have to tune in a year or two.
Right, maybe give me free.
Let's switch spaces a little. So now let's think about you as a teacher. What school are you part of at Rutgers, and which department, and how are you contributing to those activities?
Yeah, so I'm in the Robert Wood Johnson Medical School, and I have a primary appointment in the Department of Pharmacology with a secondary appointment in the Department of Medicine. And so my first year, so as I'm new here, my first year has mostly been protected research time. But in the coming years, I expect to integrate a bit more with the department and hopefully be involved in lecturing and mentoring students in the graduate courses and the medical students in pharmacology subjects.
Yeah, and speaking of students, you're just setting up your lab. So maybe you can tell us, and of course you want to recruit people to work with you, students and postdocs. So how do you interact with students? And trainees, what's your philosophy?
Yeah, so I've been very lucky so far. I think at Rutgers we have a wealth of really engaged and engaging undergraduates, actually. So we have a couple of undergraduates in the lab already, and they come in on on a week-to-week basis and really are learning new skills in the lab in molecular biology and some of the animal techniques that we do. So they're really contributing to the lab. In addition to that, we have 2 postdocs in the lab. So I have Vinny, who joined about a year ago. She's an electrophysiologist. And then Kingsley is a cardiovascular physiologist who joined a few months later. And so the lab is building up, and we have all of these people getting busy with experiments, learning new techniques, and also sharing their techniques with each other, which is a really satisfying thing to see. So this is an exciting time when we're building things up, and I'm trying to keep things going. So yeah, we're making it up as we go along at this point.
Perfect. And so, which is, by the way, what we all do.
Right.
At every point. So now I want to just finish with a question that may seem a little weird because you're a young guy. And I want to ask you, thinking about what you're doing now and kind of the directions you want your career to take. Let's say that unfortunately, years later, somebody has to write your scientific obituary, and they have to kind of summarize, what's the most important thing you did, or your most important contribution. What do you what would you like that to be?
Right. I think if in this hypothetical, if someone's writing this about me, then something's gone right. So that's good
right.
news.
It's not the young you, it's the
right.
old you.
Right. So hopefully that means I was able to continue doing this job for a long time, which is one main objective, of course. So yeah, so hopefully what we are able to produce in the coming years has some impact. Maybe we can provide some physiological mechanisms, some insights into basic biology. And then, of course, the overarching long-term objective is to try and find some scientific advances which can translate into improving human health. So that's the long-term objective. But really, on a day-to-day basis, what I'm fascinated about is experiments, right? So thinking of a new idea, finding a way to test it in the lab, and hopefully it's an elegant way of testing it in the lab that gives a clear result that we can then tell other people about. So that's really that's the motivation on a day-to-day basis, but hopefully it sums into something that has a wider impact in the world in the future. And then, of course, the final thing would be hopefully with time, I will have the chance to mentor other scientists and to provide them a pathway to express themselves scientifically and develop their own careers.
Oh, beautiful. I think that's a full career there. So,
right.
It's funny because, sometimes the great discoveries are accidental. So that first syndrome that you mentioned, which leads to excessive hair growth, people might think, well, how can we capture that particular phenotype and use it to our advantage?
That's very astute you say that, Marty, because the genes responsible code for proteins which are the target for topical alopecia treatments, such as Rogaine. Yeah. So there is some link there. So studying these rare diseases can provide insights into broader physiology that can be applied elsewhere, that's for sure.
Anytime you have a phenotype, you can discover something.
Right.
Conor, it is terrific to speak with you, and I wish you luck in your career and that you will soon fill up all your shelves. And thank you for coming to CABM.
Great. Thank you very much, Marty. Thank you for all the support so far. It's been a pleasure.